So I'm very honored to be chair of the scientific committee of this 33rd ESMO Congress. I would like to give you perhaps a few key messages about the scientific content of this meeting. I think what we realize when we come to well-organized meetings like this one is how important clinical research is to improve the care of cancer patients. And clearly, we are very fortunate at this meeting that for the first time, results of some large clinical trials will be presented. And these results have the potential to indeed change practice and encourage physicians to prescribe better treatments for their patients. Now, you should know that these clinical trials are extremely difficult to run today. They require a lot of energy. They are insufficiently supported. And from time to time, what we see is that we lose fantastic opportunities because today we have great technologies that should enhance our ability to understand why some patients respond beautifully to a cancer treatment, while other patients have absolutely no benefit. So now we have the tools to analyze the tumors of these patients and start understanding what is the explanation behind this differential response to treatment. Up to now, we have had very few tools as oncologists to identify upfront the patient who is going to benefit from a treatment from the patient who is not going to benefit. So what we do is we prescribe the treatment, and after a few months, we then suddenly realize that the treatment was the right one or was not the right one. So what is clearly becoming very important, and we see it at this meeting, is the translational research that is linked to the clinical trial. So today when we run these clinical trials, we do all the possible efforts to collect the tumors of all the patients, to collect some blood, and start to analyze the genetic background of the patient, for example, because we start to see now that this can tell us, indeed, who is going to benefit from a treatment and who is not going to benefit. We are also collecting the tumors because we have tools to analyze the genes that are active or inactive in the tumor, and that turns out to be a very important information about the chance to respond to the treatment. So an important message is that we need to invest in these clinical trials. They are going to be expensive, but at the end of the day, we will be in a situation where we will be able to prescribe much better therapies to the right patient populations. So let me give you one example of what I think is an important trial which is going to be presented at this meeting. And interestingly, it's a trial that has been conducted in Asia. It's a trial conducted in patients with advanced lung cancer. You know that lung cancer is a big killer. The particularity of this trial is that it was restricted to patients who had never smoked or were previously light smokers. And the trial confronted the standard of care for this disease, which is chemotherapy, with all its side effects, to a targeted treatment, a clever treatment, which in fact is a molecule that can interfere with a growth factor receptor. So to explain to you what I mean by that, tumors are highly dependent on growth factors that exist in the surrounding environment of the tumor. To exert their action, these growth factors need to recognize a receptor that is on the cancer cell. The receptor is like a key. So the growth factor comes into the key and turns the key and can then send a message of growth to the cancer cell. We now have very nice targeted compounds, drugs, that can block the key. And in this trial, the investigational treatment was one of these very smart drugs that can block the key for cancer growth. And the findings are really impressive because what turned out to be the better treatment was indeed the new treatment. Not chemotherapy, but this very smart drug, which was able to shrink the tumor more often and to reduce the likelihood that the tumor would progress. And of course, there was also a better quality of life for these patients because the side effects of this smart molecule are less than the side effects of chemotherapy, which is not a very selective treatment. So this trial, I think, is important because it gives us the message that with what is happening today in oncology, it's clear that we are moving to a new era. In a few years from now, I think we will be prescribing more and more this kind of targeted treatment. We will be able to better identify the patients who can benefit from these treatments. So that is, I think, one of the most important take-home messages from this Congress. And so I think I would be delighted if you would somehow also give the message to the public that governments have to invest in this kind of research because it's the only way to improve, first of all, the treatment of cancer patients, but it's the only way to cut the cost also to some extent because all these therapies are very expensive. And if we do not do the right research at the beginning and identify the patients who are really helped by these new drugs, we will have to pay for these drugs for everybody, and that is becoming a very high financial cost to society. I think I stop there and see if there are questions, if I need to clarify certain points, or if you think it was clear. The drug I'm talking about is gefitinib. So it's one of these very small molecules that blocks the growth factor receptor. It's also called, the name is Iressa, the commercial name. It's a drug that has been known to be effective in lung cancer for some time, but this is one of the first trials where it has been used as a single agent, not combined with chemotherapy, and as the first treatment for these patients. It's a study with a very original design, so it is the confrontation between a very rather aggressive treatment, chemotherapy, that works in approximately 1/4 of the patients, because lung cancer is not a highly chemosensitive disease. And it's a comparison between this treatment that we've been using until now with a targeted drug, a drug from the new era, that really targets a specific characteristic of the cancer cell. And what I think is very striking here is that you would have expected perhaps that it would be slightly less active or similarly active to chemotherapy. It turns out to be more active than chemotherapy. And that's probably because the patient selection was also right. So what it tells us is that if in the future we are able to select the right patients for these very intelligent drugs, we will improve the care of patients significantly because we will use drugs which have less side effects than chemotherapy.
Just probably to add to this story, this drug has been proven in combination with the same chemotherapy, which was proven here as single agent or as without gefitinib 5-6 years ago, 7 years ago. And it failed. It was not better than the chemotherapy in combination. So then was a question, why this is the case, why it's not more active. Also, there's some single agent activity for this gefitinib drug. And now we have learned that there was a mixture of patients, smokers and non-smokers. And this study is new, just taking non-smokers or very light ex-smokers. So this is the target population for Iressa, for this gefitinib, for the EGFR tyrosine kinase inhibitor. And if you purify the population of patients, and this has been shown here, then you get results, not in combination, because the combination has antagonistic effects in this disease. But as single agent, it's very active and even more active than the standard chemotherapy. So that is a new part of this. As combination, it failed completely. Many trials, two large trials with thousands of patients completely failed. So now we learn how to use these new drugs better to be successful for the patients.